Hey guys, I'm back. We're going to talk about enzymes and particularly focusing on factors that could influence how well an enzyme functions. So the five factors that we're going to focus on today are enzyme concentration, so how much enzyme is present, substrate concentration, how much substrate is present, and then temperature, pH, and salinity. There are other factors that might influence how well an enzyme functions, such as inhibitors. So those would inhibit the functioning of the enzyme. But these are the ones that we're going to focus on for today. So let's start with enzyme concentration. So on this graph, we're looking at how increasing the amount of enzyme present affects the reaction rate. So how quickly you go from substrate to products. So let's take a look at this. What do we see happening? So you have this increase in reaction rate as you increase the amount of enzyme, and then eventually you flatten out. So what's happened here is you've kind of reached the maximum reaction rate at that particular concentration. So that enzyme is working at its optimal rate here. It can't go any faster. Generally, that's because all of the enzyme is interacting with the substrate. So if you think about it like this, as you increase the amount of enzyme, you're going to increase the reaction rate because the more enzymes there are, the more frequently they interact or collide with the substrate. However, at some point it's going to level off, which means that the substrate has become that limiting factor, the thing that's preventing you from going any faster. So at that point, there's enzyme molecules that are just hanging out because there's not any substrate for them to react with. When we graph the effect of increasing substrate concentration, so how much substrate is present, and with keeping the enzyme constant, you see a very similar pattern. And the same kind of thing is going to be happening where you reach that maximum reaction rate where the amount of enzyme becomes limiting. We say that the enzyme is saturated. That's its maximum rate. So the only way to make it go any faster would be, again, to increase the amount of enzyme here. So for both enzyme and substrate concentration, you have this increase in reaction rate as you increase the concentration until the point where you've saturated or you've reached the kind of maximum rate. All right, here we have temperature. So let's take a look at what's happening in this particular graph. So this looks a little bit different, right? So we have increasing temperature and we have the reaction rate again. So how quickly are you breaking down whatever your substrate is into products. And here at 37 degrees on this graph, we have it increasing until it peaks here, and then all of a sudden it starts to decrease. Now 37 degrees Celsius is actually going to be body temperature. And for many enzymes, particularly human enzymes, that's actually going to be the optimal temperature. So we call this point where you have that maximum reaction rate the optimum temperature. And so what happens is at a low temperature, molecules are moving slower. And so they're less likely to collide with the enzyme. So enzyme and substrate are less likely to bump into each other. So if you imagine just walking really slowly through the halls, you're less likely to bump into someone. If you start jogging through the halls or running through the halls, you're more likely to bump into someone. There's more movement, there's more energy in the system. And so as you're increasing that movement, the likelihood of your enzyme and substrate to collide and react is increasing. However, at a certain point, something starts happening to that protein or that enzyme. It depends on those hydrogen bonds and ionic bonds to hold it in its three-dimensional shape, that tertiary structure. But if there's too much energy that starts to fall apart. It starts to break. And when that happens, we say that the protein has been denatured. So when you're cooking your food, a lot of what you're doing is denaturing the proteins, which makes the protein structure change. That's why, like when you cook eggs, they go from clear to white, or the white part does. You've denatured those proteins. So at that point, the structure doesn't fit, and so it can no longer catalyze the reaction. Now, not all enzymes are going to have 37 degrees as their optimal temperature. If you go and look at, say, bacteria that live very happily in these hot springs, we found that they have enzymes that have an optimal temperature around 70 degrees Celsius. So really different organisms and different enzymes have different optimal temperatures. So now let's think a little bit about what tools you would need to measure and adjust temperature in a lab setting. Well, if you want to measure temperature, you're going to use a thermometer, and you're always going to use Celsius. Now, if you want to heat something up, you'd use a hot plate or a hot water bath. Now, if you're at home and you're doing that, a hot water bath is basically water that you've heated up in a container. So it's a nice little bath for your reaction. Generally, when you do this in a lab, you have a beaker full of water and you heat it up. Or there's actually something called a hot water bath that will heat to a particular temperature. Got one of those fancy sous vide machines. That's usually what they do. They heat to a certain temperature. And you can utilize that to maintain the temperature of your reaction. What if you want to make it colder? You do know the answers to this. It's there. So you just use ice or your refrigerator. Those are great tools for making something colder than room temperature. So these are different ways that you could measure and then adjust temperature in a lab kind of setting. All right, here's another one, pH. So remember your pH scale, 7, lucky 7 is neutral. Anything on this side over here, this 1 to 7 or 0 to 7 would be considered an acid. Anything above 7 is going to be considered a base. The closer to the extremes or the further away from 7 you are, the stronger that is. So something with a pH of 1 is a stronger acid than, say, a pH of 4. They'd both be considered acids, though. So here, we end up seeing very similar kind of increasing until you get to that point and then decreasing outside of it. Now notice that this is a really even distribution on either side, whereas with temperature you had this kind of, so if it was temperature it would be like increasing and then a sharper drop off, but for pH you have this even one. Just keep that in mind if you ever have to draw that, and I'm going to erase that. All right. So for pH, again, you see a pretty similar pattern, though, where you have this optimal point where the enzyme works really, really well, but outside of that range, it stops working. And this is really interesting. So if you look at enzymes, particularly in humans, the ones found in your digestive tract, the stomach is at a very, very acidic pH, so a low pH. And then your small intestine actually is going to have a more basic pH. So enzymes that function in the stomach are going to have an optimal pH that's much lower, like say something like pepsin. And then enzymes that are found in the small intestine, like trypsin, are going to have a higher pH. And that's also a way of controlling when and where they're functional. So just to kind of understand how that works on a chemical level, when you change the pH, you're basically adding or removing hydrogen ions. And those hydrogen ions can disrupt the bonds. Again, you have those attractions between the charged amino acids in your secondary and tertiary structures. And so if you are disrupting those bonds, you can change the shape and denature the protein. And remember, proteins depend on their structure to function. So if you change the shape of the enzyme, you change its function. So like I said before, most human enzymes function best at that neutral pH, but there are exceptions, particularly in the digestive system. So this is another one that comes up. So if you're trying to conduct a lab experiment and you want to actually adjust the pH, there's some different ways that you can do that. In a lab setting, we use something called a buffer, and this will help maintain your pH within a certain range. And then you can use good old-fashioned pH paper to measure the pH. There's also something called a pH meter if you need more specific data for that. If you're at home, there's some interesting things that you can do. You can actually make pH indicators using like red cabbage or I want to say radishes do it too. There's a few different vegetables that you can actually boil, and they have natural pH indicators in them. So basically, these are chemicals that will change color in the presence of different pHs. Sometimes you'll notice that when you're like doing the dishes and you've cooked with that kind of food. You also can find some pH supplies. If you have a fish tank, you may have something that will help you measure the pH. In terms of adjusting, there are some household ingredients that could help you adjust the pH of your solution, but you do have to keep in mind that those come with their own kind of limitations, right? So if you're adding lemon juice and you're adding ammonia, you're also adding substances that might be influencing the reaction outside of the fact that they're changing the pH. So there's some limitations to that, but there are ways that you can do these things at home. And the last one is going to be salinity, or the last one we're going to look at. And here again, notice you don't have that very even, but you have this kind of increasing, and then dropping off. You have these optimal levels. And here again, you're adding or removing positive and negative charges, and you're disrupting the three-dimensional shape. So if you change the shape, you change the function, and so you will denature the protein. Most enzymes are going to be pretty intolerant of that extreme salinity. Again, you're disrupting its structure, so it can't function. The Dead Sea is a really good example of an area that does not have a lot of life because there are not a lot of halophiles that can survive in that environment. And again, just trying to think about if you were to adjust this in a lab, how would you actually do that? How would you measure it? Well, you could use good old-fashioned table salt, NaCl, sodium chloride. You'd actually want to use the cheaper table salt that doesn't have any iodine in it. Most of the salt at the grocery store is iodized, meaning there's trace amounts of iodine in it. But the cheaper salt actually doesn't have that in it. What kind of units? Well, we talked about this when we were looking at the concentration for the last lab. You could do all sorts of ways. You could use different ways of measuring concentration. So you could do moles per liter, so molarity. You could do a percent concentration. You could do grams per liter. Any of those would be appropriate. The major thing to think about is like, why are you using that level and why do you think it would be important? So hopefully that was helpful to you. Make sure that you can answer the questions posted and think about different labs that could kind of go along with it. How would you actually do these things if you were trying to do it in a lab setting?